Hello everyone, we are now at a very important part of our lecture series on overview and integration of cellular metabolism, where we will be starting with metabolism of individual amino acids. Today's class will be discussing metabolism of phenylalanine along with its associated disorders, alright. So, we will be mainly discussing regarding the whole metabolism of phenylalanine. We will be focusing on the disease that is characterized by disorder of phenylalanine metabolism, that is phenylketonuria. We will be discussing its etiology, the various variations of phenylketonuria, how it is inherited, what is the incidence of phenylketonuria, the various clinical features, diagnosis and treatment. We will be covering it all, alright. So, here is a chart about all the essential and non-essential as well as conditionally essential amino acids. This is the total 20 in number, alright, and among them we have chosen to start with this amino acid phenylalanine, alright. Do you have any idea why? You will get the answer very soon, but today's class we will be discussing phenylalanine metabolism. Mind it, this chart will be essential for you to consider all the essential, conditional essential as well as non-essential amino acid for multiple choice question purposes. So, it is very important you at least know few example of essential, non-essential as well as conditionally essential amino acids. Okay. So, phenylalanine, this is the structure of phenylalanine, alright. We all know whenever we are discussing an amino acid, there is a carbon skeleton, right, there is a carbon central carbon atom and there is a COOH group attached to it, there is an amino group, there is a hydrogen, and this is the part that varies which isolates each amino acid from another. So, in case of phenylalanine, it is a benzene ring along with a CH2 group that is attached to this carbon atom, right. Remember, the simplest amino acid is glycine, but this is simply an hydrogen molecule. So, this is a aromatic amino acid, alright, because there is a presence of an aromatic benzene ring. Again, essential amino acid means there is no synthesizing phenylalanine in our body. We must have phenylalanine from diet. Mind it, whenever we are discussing an essential amino acid, you do not need to read about synthesis because they are not synthesized in our body. Whenever we are reading a non-essential amino acid, synthesis of that amino acid is must, right, for your exam purposes. The metabolic fate, this being both glucogenic as well as ketogenic. When we will read phenylalanine metabolism, when we will complete the degradation pathway of phenylalanine, we will see that it is converted to acetoacetyl coenzyme A as well as fumarate. This is the ketogenic part and this is the glucogenic part, alright. However, the beauty of it is phenylalanine is first converted to another amino acid, that is tyrosine. Since phenylalanine is converted to tyrosine in our body, of course, you have known, you can sense it by now that tyrosine is not an essential amino acid. Means if we have got enough amount of phenylalanine in diet, tyrosine will automatically be synthesized. And the reverse is also true. Means the amount of phenylalanine required in diet will be reduced if there is adequate amount of tyrosine in diet. This is known as pairing action of tyrosine on phenylalanine. Means if we have got adequate tyrosine in diet, the amount of phenylalanine required is less, and this is very important, alright. So, as I told you, predominant metabolism of phenylalanine occurs through tyrosine, means first phenylalanine is converted to tyrosine and thereafter tyrosine is degraded, alright. And not only it is degraded, from tyrosine multiple very important compounds are obtained in our body. To name a few, catecholamines, that is epinephrine, norepinephrine, adrenaline, noradrenaline, neurotransmitters like dopamine, thyroid hormones, the pigment melanin, all are important products that are derived from tyrosine. So, indirectly they are derived from phenylalanine also, right. So, when this is our metabolic goal, right, mind it, whenever we complete any amino acid metabolism lecture series in this session, we will be correlating our progress with this figure. In today, this is where our goal lies. Phenylalanine, it is converted to acetoacetyl CoA, and here it is phenylalanine, it is converted to fumarate, alright. Mind it, tyrosine also goes hand in hand with phenylalanine, but and this is the total metabolism of phenylalanine and tyrosine. Basically, it is the same, the whole metabolism of phenylalanine tyrosine only varies in the first extra step where phenylalanine is converted to tyrosine, and in today's class we will be focusing only on this step, right. It is, so our goal is clear, phenylalanine is getting converted to tyrosine. If there is a problem, there will be disorder. So this will be our today's discussion, right. So let us elaborate this single step in detail, but don't worry, the entire step will be covered in future classes when tyrosine metabolism will be discussed, alright. So how exactly, what is this reaction? If we look into this reaction in detail, this is simply a hydroxylation. Tyrosine is nothing but para hydroxyphenylalanine. An OH group is attached to the para position. From your aromatic organic chemistry knowledge, you already know what is ortho, what is meta and what is para position, right. I am not going to detail. So phenylalanine is hydroxylated at the para position by an atom of oxygen to produce OH. So this is already H is there, right. So an O atom will get inside, right. And what happens to another atom of the oxygen? It is reduced to water. So oxygen gets in and water comes out, alright. But once initially it was thought that oxygen is simply going inside, right. In reality, what happens, the hydrogen atom that is shaded in pink is transferred directly from C3 to C4, okay. By carbon labeling it was shown by the National Institute of Health, and hence this shift is known as NIH shift. The hydrogen which is originally over here actually shifts over here, okay. And one hydrogen from the adjacent site actually goes to the fourth position. Anyway, so that is not our detailed organic chemistry, we do not need to go into that much detail. What we need to know, the enzyme that is causing is known as phenylalanine hydroxylase, and that enzyme requires a coenzyme, right, which is biopterin. Biopterin in the form of tetrahydrobiopterin. This is very much similar in structure to folic acid, okay. It is a vitamin, folic acid is a vitamin, and this tetrahydrobiopterin is very much similar in structure to folic acid. So this is the enzyme phenylalanine hydroxylase, and this is the coenzyme tetrahydrobiopterin, right. What happens when the reaction actually happens, the active form of tetrahydrobiopterin, it is oxidized to dihydrobiopterin, which is basically in quinoid form. Thereafter dihydrobiopterin is acted upon by reductase, and thus tetrahydrobiopterin is regenerated, okay. So you can see over here, it is oxidized and then it is again reduced, right, and this phenylalanine hydroxylase enzyme is present in liver. Well, there is also another intermediate, if we really want to learn in detail, this is the actual form where there is a carbinolamine derivative that is also formed, right. So in reality, in order to regenerate the tetrahydrobiopterin, two enzymes are playing role. One is carbinolamine dehydratase and one is dihydropteridine reductase, right. Mind it, the name of the enzyme is dihydropteridine reductase, is often termed as dihydrobiopterin reductase. It goes hand in hand, more or less the same thing, right. So phenylalanine hydroxylase is the enzyme, tetrahydrobiopterin is the coenzyme, this is the active form, it gets reduced and ultimately it again gets regenerated, and these are the intermediary enzymes that are all acting together to catalyze this one single step of conversion of phenylalanine to tyrosine, alright. So now whenever we are discussing, so our one step, that is goal of today, is done. So what we will be discussing now on is what if this step is hampered, alright. So in general, whenever we are dealing with any inborn error of metabolism, whenever there is loss in enzyme due to either deficiency or loss in genetic mutation, whatever be the reason, what happens? Generally enzyme reacts on substrates in the presence of cofactor and it forms product. Whenever enzyme or in some cases even the coenzymes are having a problem, this step will not be happening, which will lead to excess accumulation of substrate, and naturally there will be depletion of the product. This is a phenomena of every enzyme deficiency, alright, and we have already read a few regarding ammonia metabolism, right. So, enzyme deficiency of urea cycle, the same concept holds there also. Any enzyme defect, the substrate accumulated, the product deficient, right. So, the disease that happens when this enzyme phenylalanine hydroxylase is deficient is known as phenylketonuria, which is also abbreviated as PKU. Okay. So, this is the most common disease of amino acid metabolism, and that is the reason why we are studying, why this course has been designed in such a way, so that you need to know the worst disease at first. This is the most common amino acid metabolism. Hence, we have chosen phenylalanine metabolism to start with. Now, you know the answer. This deficiency due to phenylalanine hydroxylase enzyme, multiple choice question, where what is the gene that codes for this enzyme? PAH gene, located in the long arm of chromosome 12 and locus 22. Another MCQ question, very important. You should note it down in your separate notebook if you do have a habit of making your own notes. Inheritance via autosomal recessive mode, and what is the result? The substrate will accumulate, phenylalanine cannot be converted to tyrosine, phenylalaninamia, hyperphenylalaninamia, alright. So, when phenylalanine hydroxylase is deficient, phenylalanine will be accumulated, there is no problem in understanding this, but even if phenylalanine hydroxylase is fine, but somehow the coenzyme is having a problem, the level of coenzyme is not functioning or this is not present in active form, then also this reaction will not happen and phenylalanine will be accumulated, alright. I hope there is no problem in understanding that. Anywhere problem in either enzyme or coenzyme will lead to disease process, and we have already read that these are the intermediary enzyme that actually help in regeneration of active form of tetrahydrobiopterin, right. So, this is dihydropteridine. There is also another enzyme which is dihydropteridine or dihydrobiopterin synthetase, DHBS. Even if that enzyme is deficient, it indirectly leads to accumulation of phenylalanine, because all these three enzymes if deficient, this leads to depletion of coenzyme and hence this reaction cannot proceed, right. So, based on all of these etiologies, the phenylketonuria can be classified into classical phenylketonuria, which is the most common, most variant variety, and it is characterized by deficiency of the enzyme phenylalanine hydroxylase. However, there are atypical causes of hyperphenylalanemia, right, due to deficiency of the coenzyme, that is tetrahydrobiopterin, and these are the intermediary enzymes. Any one's deficiency may lead to a atypical hyperphenylalanemia. Mind it, whenever we are referring to phenylketonuria, for all practical purpose it refers to the deficiency of phenylalanine hydroxylase and classical phenylketonuria. These are all atypical and often these are not termed as phenylketonuria, okay. But nevertheless, these are all phenylketonuria variants, they are atypical, they cause hyperphenylalanemia. So, regarding the altered metabolism of phenylketonuria, now we are all in the same page, because either if the enzyme is deficient or if the coenzyme is deficient, phenylalanine will be accumulated and there will be no production of the product with, that is tyrosine. I hope you are fine with this, right. So, in absence of phenylalanine or either the enzyme or the coenzyme, phenylalanine cannot be converted to tyrosine. So, what is the problem? In the beginning I told you, it is from tyrosine where multiple important products are formed. So, one by one we will be seeing what are the, we will be naming here what are the products that are formed, and that will be a problem in case of phenylketonuria, because the downstream metabolism of tyrosine is fully blocked, the downstream synthesis of important product from tyrosine is blocked, and one of them is melanin. What melanin does? Melanin helps to impart a color, blackish or brownish or reddish color to our skin. It protects us from sun, okay, the harmful UV rays. If melanin is deficient, that will lead to conditions like fair skin, light skin, albinism, vitiligo. There are multiple such disorders which we will be discussing in detail when we are discussing melanin synthesis from tyrosine. But for now, the common sense, as you can rightly see, tyrosine is not formed, hence melanin level is much less, that leads to light skin in phenylketonuria patient. Moreover, the enzyme, the rate limiting enzyme, one of the most important enzyme in melanin synthesis is tyrosinase, alright. Tyrosinase is inhibited whenever phenylalanine concentration is high. So, all in all melanin synthesis is hampered, and that leads to light skin in case of patients of phenylketonuria. Not only melanin, what happens from tyrosine, synthesis of catecholamine happens. So, if catecholamine means adrenaline, noradrenaline, they are very important neurotransmitters, okay. They are very important mediators of multiple reaction. Not only that, if the coenzyme is at fault, the coenzyme tetrahydrobiopterin is also involved in tryptophan metabolism. Again, it will be taught to you in detail, where tryptophan gives rise to a very important neurotransmitter, that is serotonin. So, you see, either the enzyme deficiency whenever tyrosine is not formed, or even if there is deficiency in the coenzyme tetrahydrobiopterin, it will lead to a situation where important neurotransmitters like catecholamine, serotonin are absent, are produced in much less than required in our body, and which will lead to problems, right. Next, see, phenylalanine, what happens if it cannot be converted to tyrosine, right. So, naturally phenylalanine level is very high, okay, in blood. These are essential amino acid. So, brain has got, and they have got important requirement in brain for development in normal cases. So, phenylalanine, what it does, it is a large neutral amino acid, and it is normally transported into the brain through the blood brain barrier with the help of large neutral amino acid transporter, okay. So, which it is fine, but if phenylalanine is excess in blood, it will saturate the transporter. So, whatever transporter is there, all will be attached to phenylalanine, because it is much excess, all will be binding to that ligand. And what will happen? Other neutral amino acid, other long neutral amino acid cannot get inside the brain. Those long neutral amino acid are extremely necessary for proteins and other neurotransmitter synthesis in brain, and that will be hampered if excess phenylalanine is present in blood, which ultimately results in hampering of brain development, especially in the growing period, because mind it, if all these deficiencies are congenital, right, these are all inborn error of metabolism. So, when there is an increased requirement of essential amino acid from brain development, those will not be found, and hence hampering in proper brain development will lead to intellectual disabilities, right. So, this is one thing. So, phenylalanine is not getting converted to tyrosine. So, what is actually happening? Phenylalanine will always search to be metabolized in some way once something is being accumulated. So, in our body, phenylalanine is mediated through alternate routes. These are conversion to phenyl pyruvate, phenyl acetate and phenyl lactate. So, be it transamination, be it decarboxylation, ultimately it leads to conversion of phenylalanine, because it is present in very high, into unwanted compounds which are normally produced in very less or negligible amount. But in this case, in case of phenylketonuria, these are produced in excess, and all of these have got characteristic properties, and this are also active agents. If accumulated, they also have been found to be culprit in brain damage, right. So, with all these altered metabolisms, if we now dissect the symptoms of phenylketonuria, it will be very clear for you to guess, alright. So, first CNS symptoms, central nervous system symptoms, mental retardation, hyperactivity, jerky movement, failure to walk, talk, acidity, abnormal movement like athetosis, seizure. Why? Because there is a problem in neurotransmitter, essential amino acid, etc. Okay. So, all of that leads to poor brain development, intellectual disability. So, all of them culminates into CNS symptoms, which is the major dreaded sequence of phenylketonuria if it is left undiagnosed. Okay. Next, hypopigmentation we already discussed because of decreased amount of melanin, and mousy musty odor. A phenylketonuria patient, the urine, the secretion, the body fluids have got a typical odor, musty odor or a mousy odor, okay, that is actually found from surface of rodents. Why that is found? Because of presence of excess phenylactate, phenylacetate and phenylpyruvate, those alternative compounds that are produced in minor pathways from excess phenylalanine get secreted into the body fluids, breath, skin and urine, that leads to a mousy smell, right. So, how this phenylketonuria is inherited? Phenylketonuria, now we are discussing about the heredity. As I have already discussed, this mutation in PAH gene is an autosomal recessive condition, means both parent can be, so the gene needs to be homozygous in order to express, right. So, both copies need to be homozygous. So, if there is only a single copy, a man or a woman will be a carrier. So, a carrier dad and mom, if they conceive, you know, these are simple Mendel's law, one of them, that is 25% of each pregnancy will be affected if both are carriers, alright, and one of the babies will be non-carrier, alright, and 50% will be carriers. This is simple Mendelian genetics, okay. So, how can we diagnose phenylketonuria? This diagnosis of PK is extremely important, because if we fail to diagnose, there will be dreaded consequence of brain damage and intellectual disability, which nobody should deserve, right. So, naturally prenatal diagnosis, even before the baby is born, we can diagnose phenylketonuria prenatally. How we will have an idea that the baby can have, if there is history of carrier, if the maternal or any of the parent is a carrier, or preferably both are carriers, then the situation is almost dreaded. It can happen, right. So, prenatal diagnosis in fetus by detection of gene mutation, PH gene mutation. If it is mutated, then there is a chance that the baby, or there is a 100% chance that the baby will be phenylketonuria. But mind it, as I always discussed previously, a mutation may lead to total or partial loss of the enzyme. So, how the symptom happens is varies from individual to individual. Next, after the baby is born, neonatal diagnosis, how it is done? By assessing blood phenylalanine level. Normally, phenylalanine level is about 1 mg per dl, it is very low, but in case of phenylketonuria, it is above 30 mg per dl. It has to be above 30 mg per dl, but in milder cases it has also been found from 6, as low as 6. I mean, cases of phenylketonuria with PH gene mutation, 6 mg per dl, 6 to 30, those are milder condition, but generally if there is a defect, it is usually found to be above 30 mg per dl. So, there is a huge difference between the normal level and the raised level. So, once the level is raised, there is no second thought in detection or diagnosing that this is the case of phenylketonuria. So, how a sample is collected in those small babies, because these are all inborn error of metabolism, right. A neonate, small babies are the patients from which samples need to be collected. So, basically a few drops of blood is obtained by heel prick, alright, and these are soaked on cards. The customized cards are available which can actually diagnose or collect. We can either collect blood in blotting paper, or cards are available in which the bloods are soaked, and then these cards are actually sent to the lab for our newborn screening, because generally there have been cases where the symptoms overlap, and one single mutation has been found, multiple mutation can also be found. So, a child may be affected with multiple amino acid inborn error of metabolism. So, in general there is a newborn screening is done where one by one all the amino acid deficiencies or all the amino acid enzyme deficiencies are checked. So, this is the way how sample is collected, I mean blood sample is collected by heel prick, card soaked on card and sent for easy identification and early identification and implementation of prompt implementation of treatment, right. So, what is the method of choice? So, nowadays all inborn error of metabolism, if you get the MCQ this option, you should always tick it, that is tandem mass spectrometry or MSMS. Any intermediate substrate or product or deficiency or metabolite needs to be diagnosed. In this case, we are trying to diagnose the phenylalanine level is done by MSMS or tandem mass spectrometry. This is a schematic diagram of mass spectrometry, and this is beyond the scope of this class to discuss how mass spectrometry happens, but you can always enroll into other NPTEL biophysics classes to get your concepts clear about mass spectrometry. All right. So, now we discuss about some tests that are very convenient now to perform, but more nowadays we are not using them. So, these in near future will have historical importance, but nevertheless these are very important to know. So, what is Guthrie test, right? This was one of the earliest tests that was devised for phenylketonuria. It can be done in simple laboratory setting. So, what happens? Blood, the sample is plated on an agar plate, alright. Agar is a culture media and it contains a special type of bacteria that needs phenylalanine, that is essential amino acid to grow. So, normally the media is devised in such a way that there is no phenylalanine or very less phenylalanine for the bacteria to survive and grow. But if the patient's blood have got, I mean if the patient is of phenylketonuria, his blood level of phenylalanine will be so high that the bacteria will find it very conducive to grow. So, if there is bacterial growth, phenylketonuria is positive. So, basically let us see what is happening in a figure. So, this is the culture plate where there are positive blood. So, all the 4 types are usually placed. So, blood is placed in samples in dot fashion. So, you can see there are positive controls where increasing phenylalanine concentration are provided to see how much the bacteria is growing. So, more phenylalanine means more bacteria will grow and there will be a halo around the spot, the haze, it signifies bacterial growth. And if there is no negative, I mean the negative control means there is no phenylalanine. So, we have got something to compare to, right. In case of the sample, you see over here it is showing a halo which can be compared to the positive control, right. So, this case, this is a patient where this patient has shown a positive phenylketonuria. Minded, these are all samples for individual patients, right. So, in this case all of them have showed negative growth, but this has been shown a positive growth, and we can surely say that the phenylalanine level is very high and the case is strongly suspected to be of phenylketonuria. All right. Next, there is another inexpensive test, that is ferric chloride test. It is generally done in urine. So, what it does, how the test is performed? Freshly collected urine sample is diluted with ethanol and few drops of freshly prepared ferric chloride is added to it. Fresh urine sample, fresh ferric chloride. So, what will happen? Phenyl pyruvate, being a phenyl ketone, actually gives an intense blue green color and it indicates a positive test. So, if there is phenylalanine, ultimately it will be converted to phenyl pyruvate, okay, alternative paths of metabolism, and that will lead to phenylketonuria positive test. So, negative, if the test is negative, it will show brownish or yellowish color of ferric chloride. Okay. So, positive test for ketone bodies is, test for ketone bodies is positive in phenylketonuria. It is a very important trivia. Remember, when you are dealing with practical exams, generally we have discussed when ketone bodies are present during prolonged starvation, diabetes mellitus. We have all read ketone body metabolism in our lecture series, and we have discussed that Rothera's test is positive in that case. However, Rothera's test is also positive in presence of this disease phenylketonuria, because phenyl ketone gives a positive Rother's test. Okay. Now, let us come to the incidence of phenylketonuria. If we sort it by country, Turkey, these are 2017 data, Turkey has been shown to have the highest incidence of phenylketonuria, and if we compare it with India, we are somehow in between, we are not at the top. Okay, you can see the incidence is very high in Turkey. The incidence of America is also given in this study. So, what are the precautions if we diagnose a case of phenylketonuria generally, and if the patient has grown into adulthood? There are ways by which it can be done. We should always ask them to avoid artificial sweetener, you know, sugar free, natura, all those brand names that are available in India and multiple other brand names that are available in other countries. So, that is actually derived from phenylalanine and aspartic acid, and that artificial sweetener will be a problem, because we do not want to increase the phenylalanine load of the body, because phenylalanine cannot be metabolized in case of phenylketonuria. And very important, if a woman with phenylketonuria, mind it, she is suffering from the disease and she is in a childbearing age, she should closely adhere to diets that are low in phenylalanine. If excess phenylalanine is produced, that will lead to again formation of those toxic compound, and that will increase the result of miscarriage, mental retardation of the fetus, microcephalic and other congenital heart disease. So, every time the blood throughout the pregnancy, the blood level of phenylalanine needs to be strictly monitored. So, what is the treatment of phenylketonuria? Sadly, there is no cure, there is no definitive treatment for phenylketonuria. So, generally strictly controlled or phenylalanine free diet up to the age of 14 years. All right. Permanent monitoring of blood phenylalanine level, extremely essential. After this age, generally after 14 years have passed, brain development is not that much prominent. So, extent of phenylalanine or phenylalanine then can be relaxed, some relaxation can be given in the diet, and brain is not affected by the levels of high phenylalanine in the body, because the transporter, even if it is saturated, it will not hamper in brain development. However, we should note that phenylalanine itself is an essential amino acid. So, small doses must be supplied, right, because there are products from which, I mean, phenylalanine itself is accumulated in multiple structural proteins, as it is an essential amino acid. So, small amount should be provided. Why? Because insufficient, if it is totally lack of phenylalanine diet, again it will cause mental and physical sluggishness, loss of appetite, anemia, rashes and diarrhea. All right. So, to conclude, this lecture has covered the importance of phenylalanine as well as the metabolic fate of phenylalanine, how phenylalanine metabolism is altered in phenylketonuria, what are the etiology, what is the main etiology and variations in phenylketonuria, how it is inherited, what is the incidence of phenylketonuria across countries, what are the clinical features and symptoms of phenylketonuria, what is the how we can diagnose phenylketonuria, and how phenylketonuria is treated. All right. So, these are my references and I thank you for your patient hearing.